process.
Hey everyone, we'll just get started in a couple of minutes. Today's lesson is going to be on evolution, sort of biodiversity and classification, but to the extent that would be expected at an AES standard. So I'm not going to be talking about things like the Hardy-Weinberg principle, nitrogen cycles. Basically, we'll be leaving it to kind of an AES standard of content. So biodiversity, factors affecting biodiversity calculations, the 3-domain system, mechanisms of natural selection, types of variation. And we'll basically start this plot or this story from the point where, can people hear me now?
I can hear you.
Sorry, can you hear me?
Yeah, I can hear you.
Okay, cool. Sorry about that. Yeah, we'll be starting this story kind of from the point where, where we left off in terms of mutations and meiosis, and we talked about meiosis being able to generate variation in a population. We're now going to talk about the types of variation. How does natural selection occur from a population that has lots and lots of variation within it? And then from there, we'll be building onto biodiversity. So types of variation. So we talked about in meiosis, we talked about two things that really cause variation. We said variation can be caused by crossing over of homologous chromosomes. But we also talked about variation in, due to independent assortment. So those were your two key mechanisms of variation, if you remember from meiosis. We're now going to talk about things where that definitely goes wrong. This isn't that, sorry. We're now going to talk about how that in a population level contributes to creating the variation that we see in our populations. So, for example, we all, as human beings, have variation in our populations. We don't look alike. And there are certain things and certain skills that make us adapted to do certain things. So if you think about this from just a purely human perspective, natural selection, although isn't really happening at the same rate as it's happening in other animals, natural selection is still a thing that's happening. So genetic diversity is a measure of the number of different alleles of a particular gene in a specific population. So remember, alleles are different types of genes. So a gene is something that codes for a specific protein. An allele is then a variant of a gene that codes for some other specific kind of aspect to it. And your genetic diversity is also your gene pool. It's basically the different alleles that can be passed and spread through a population. Allelic frequencies can therefore be measured by counting the number of populations and measuring within that population how many alleles or how many characteristics exist within that population. The wild type allele is the normal allele that has the highest frequency in the population. That's it. So if a population has majoritively brown eyes, then the wild type allele will be the gene that codes for those brown eyes. Remember, genetic variation is created by mutations. Now, mutations, whenever you think about them and whenever you've thought about them before, are necessarily harmful. We always think about mutations in the sense that mutations can be quite dangerous and they can cause harm to a population. But mutations don't only have to be harmful. They can be quite beneficial as well. Mutations can help a population adapt to changing environments, and that's through the natural selection process. So basically, there is a natural deviation of a population towards alleles that are going to benefit them at particular times. So another key term that you need to kind of know is gene flow. So gene flow is basically where organisms move into a population, which then causes different organisms with different characteristics to now breed. So for example, one of the big examples of this was in the squirrel population in the UK. So there used to be the red and the gray squirrel. And if you look in your parks, you'll majoritively see gray squirrels now. And what happened was within an environment, within a population, both red and gray squirrels came together, and gray squirrels outcompeted red squirrels, which meant that they were able to breed more successfully, and they were able to then add to their population. This isn't exactly gene flow. This is probably closer to competition. Gene flow happens within a species. So, for example, if you imagine like a short-necked giraffe and a long-necked giraffe, and they were both the same species of giraffe, just they had slightly different characteristics, and they both moved to an area that was kind of had high growing trees, then the tall necked giraffes are going to be able to breed more successfully. They're going to be able to breed more successfully. They're going to be able to spread that through their population. So each individual has the same types of genes as another individual of the same species. Members of a population of sexually reproducing organisms are not genetically identical. This is as they have different alleles of each gene. In asexually reproducing organisms, most members of a population of a specific species are genetically identical. This is as no meiosis occurs. Genetic diversity in asexual organisms is just through random mutations. Okay, so evolution and speciation. So evolution is the change in alleles within a population of a species, whereas speciation, and all of this happens through natural selection. Basically, natural selection is the idea that there is going to be an increase in the number of beneficial alleles due to selection pressures. Basically, things from outside the environment that benefit certain organisms and make them more able to breed. That's kind of quite the simplistic way of doing that. And speciation is the development of new and different species over time. So speciation is where new species come about by splitting the old species. And basically, this speciation occurs due to natural selection, but natural selection plus a reproductive barrier. This is really important to note, because if there was just natural selection and there wasn't anything preventing these two populations from breeding, after a couple of generations, that selection pressure will wane a little bit, and the populations would then be able to intermix again. And that would just lead to evolution within a population. But if there's a reproductive barrier, which can either be physical, it can be behavioral. So physical in the sense that two populations have split over hundreds of years, and suddenly there's a giant sea that's running right through the middle of these two populations, or a new mountain range has formed, as was found on the Galapagos Islands, which proved Darwinian evolutionary theory. Yeah, so if there's some form of barrier that's preventing these two species or these two populations from mixing, then they're only going to be able to mix within their areas. If they're only going to be mixing with the population that's around them, they're then going to split off into two different species. Okay, so three main stages of natural selection: variation, selection, and breeding. So variation step, very easy. There is variation within a population. Remember that alleles, individuals will have advantages and disadvantages to particular individuals within a species. I mean, that's common knowledge. There is going to be variation within a species. So, for example, let's look at this goat population. Some of these goats have the variant gene where they have horns produced from their head. Horns make them, there is now a selection pressure. The idea here is that some organisms are better able to mate and produce offspring. For example, in our population of goats that we were talking about here, these horns might mean that when there's a competition between two males for a breeding female, the horns on the goats with the horns is going to act as a protection mechanism. They can injure the other goats that don't have the horns. These goats then aren't able to breed because they're not able to win a competitive fight within a population. Or other examples, in the giraffe population we talked about earlier, short neck, long neck. The short neck ones aren't able to reach for the higher food, which means they die off because of that. So there is some form of selection pressure, something that is causing certain organisms to survive better than other organisms. And then the organisms with your beneficial allele then have something called a selective advantage. Basically, they're better off at breeding, which means that their genes are able to be passed on, which means over time, the number in the population that has that particular beneficial allele also increases over time, because there's just more of those kind of people with that allele, because that allele makes it easier to breed. Yeah. The thing is, there is no real human example for this. Humans don't undergo selection pressure that frequently anymore. There aren't very many selection pressures for humans, because humans are seen as the apex predator in the sense that we're top of the food chain. We literally eat everything. But if you were to think about it from a potential behavioral selective advantages, we think about people who take certain risk factors, people who smoke, for example, people who might have an excessive intake of alcohol. They have a selective disadvantage, potentially. If maybe not exactly from a evolutionary perspective, but their life expectancy is shorter. They're more likely to undergo harmful adverse effects, which means that they're more likely to have a slight selective disadvantage, if you think about it that way. And again, you can see that this population is getting more and more of this beneficial allele. All right, I don't know why the slides have broken down for a sec. Okay. But yeah, so the main rule for a selection, natural selection, is that you need to have a selection pressure. If you don't have the selection pressure that, for example, humans don't have right now, then it doesn't really matter. For example, if there is unlimited food, if some organisms are better at hunting, they're not going to out-hunt the organisms that aren't as good at hunting. A mutation can give a beneficial characteristic. This can then spread due to natural selection, and we know that. And again, this isn't one generation. This is over many, many, many generations of very small incremental population changes, because you'll have to have changes in very small number of genes. And again, if there is a barrier, if there is something splitting these populations together away from each other, then that can lead to speciation, because over time, you will have an accumulation of selective advantages for their particular environment that these two populations are in, because they're in different environments. So they're going to have different selection pressures, which means you're going to have selective advantage in a sort of divergent manner. They're going to diverge from each other, and they're going to then form a different kind of a different population species. And kind of very easy. Right, I've been asked a couple of questions in the chat. I will get to this. How can we access the site? So unfortunately, I'm not able to provide slides to you directly, but you can watch the recording back on YouTube anytime. And if you watch the recording back, they'll have all the key information that you need. Is this okay for edXLB? Again, I'm not 100% certain about edXLB as a specific specification. You'll have to check with your spec individually. This is very generic knowledge that I believe it covers a lot of biology specifications. I'm not going into specific details required for specific specs. Would you say it's a good thing that unlike other species, humans aren't evolving anymore? I think it just reflects our position. We don't have any selection pressures at the moment. I think if there was a selection pressure, humans would also have to evolve with that. And that's just something that will have to happen at some point eventually, when there is a lack of food, for example, in a population. You will need evolution to benefit you there. I think, yeah, I don't think it's necessarily a benefit. I don't think it's necessarily a disadvantage either. I think it's just simply a reflection of where we stand on an evolutionary scale. Make sense? Okay, so types of selection. So this is just about talking about different selectional types. So directional selection. It's kind of in the name, right? Directional selection is natural selection. Basically, in directional selection, rather than going for the middle ground between two kind of populations, you select for extreme alleles. For example, bacteria gaining antibiotic resistance. Directional selection, so if you imagine a normal population undergoes what we describe as normal distribution, which is kind of like a bell curve. Simple. Directional selection will have a much steeper peak at an extreme point. It's sort of a shift to the right or to the left, with much steeper peaks and lower troughs. So the overall size of the population stays the same, but there is less of a selection towards the midline of that population. And that's directional selection. Basically, antibiotic resistance is one of the examples for that, because you can either be resistant or you can't be resistant. There aren't in-betweens for it. You can't be slightly resistant, because that's just, you'll end up getting killed by the antibiotic at high enough concentrations anyway, so you're not really benefiting from that. So in antibiotic resistance, a few bacteria have the resistance, a selection pressure is applied, the resistant alleles are then selected for, and bacteria then evolve to become resistant, and a bacterial population evolves to become resistant. Stabilizing selection is the exact opposite of that. It's where we select for alleles of the mean or middle range. Human birth weight, for example, undergoes stabilizing selection, because if we have too high a birth weight, then there's harm to the mother. There's a greater risk of complications during the childbirth. If there is a low birth weight, then that can cause harms and to growth. So the individual organisms might not grow as quickly, or they might undergo harmful growth developments. So again, we want kind of somewhere in the middle, and that's why we select for that. So again, you have a very high extreme, but again, you're selecting for that midline of the graph. Genetic drift. So genetic drift is very easy to see in a small population, but this is where random alleles, random beneficial alleles are being passed on by chance. One of the best examples of genetic drift is the flu virus or the influenza virus, because genetic drift happens on an annual basis for influenza. Basically, over the course of the year, there is going to be small developments to the genetic code of the influenza virus. This small development allows that influenza virus to bypass the human's immune system. It's why you or people you know might be getting annual flu vaccines, because you have small genetic drift in very small kind of incremental changes, which allow for very small kind of populational outbreaks of the flu virus. Again, genetic drift is not really affected by benefit or harm, but overall, over time, it will be affected by the same benefit or harm, because if there's a harm for that genetic, for that small allele change, then that individual is going to die out very quickly. So over a period of time, you will notice that genetic drift will lead to natural selection every single time. And again, this mainly occurs in small population. Yeah. Genetic bottlenecking. So genetic bottlenecking is when the size of a population is dramatically reduced very quickly. And this is happening a lot. Human destruction of natural habitats. And again, I'm hoping you probably know this already. Human destruction of natural habitats is causing rapid destruction of organisms. What does that do? Well, that lowers allele variation. Which gives organisms less protection against changing environments. Because if the environment suddenly changes, they don't have the variation in alleles for them to be able to respond to that. Or they're less likely to have that. They might still have it, but they're less likely to respond to that. So genetic bottlenecking is happening quite quickly, and it's happening more frequently these days as we do things like deforestation. But again, it's a very extreme version of genetic drift. The founder effect. So the founder effect is basically where the genetic diversity in a population changes very rapidly when a small population breaks away from a larger population. If you think about it, you've got a large population with lots and lots of different alleles. A small population of that breaks away. Their variation in their alleles might be exactly the same. It might be representative of the alleles of the individual population, but it also might not be. It might be a slightly different variation of population. And again, this is the small kind of changes that lead to speciation. So for the founder effect to begin, a few individuals have to become reproductively isolated from the main group. And again, the small group are then going to be a random assortment of the population, which means that they're going to have a different set of predominant alleles that that will be present within their populations. Kind of makes sense. Does anyone have any questions about that? I'll stop for a second. Answer any questions. Adan, you've raised your hand.
I have a question. Where there was like the curves, how do they actually work? How does that distribution work?
So this is based on something called the normal distribution. So the normal distribution exists naturally. We, in human populations and in everything else, we see the normal distribution. Basically, the idea is that the extremes are less common than the mean. And it's very common. For example, let's say we took all of your school and we lined them up in order of height. You're going to have those random people that are like 6'7" and 6'8" and 6'9". But most people are going to be around 5'10", 5'11", 5'8". And there'll be a couple of people who will be very short. So the normal distribution basically plots that in a curve form. Now, when you have selection pressures, when you have stabilizing selection, basically what's happening is more people want to come towards this midline because there is a benefit in coming towards this midline, which means that your population, you'll still have some people at the extremes, but majority of people start coming towards the midline. If you have directional selection, you have more people wanting to come towards the extremes. So then you see two peaks at the extremes, and those peaks then become the majority of the individuals. So again, that's kind of the simple way of doing it. It's a mathematically derived process. So if it's very much a statistics thing. The normal distribution is just a statistics construct. Any real life examples of the founder effect? Good question. I mean, I guess you'll see it we won't see it because the founder effect is something that happens all the time. But one of the examples of it would be, yes, or lactose intolerance in Aboriginal populations. Because when the Aboriginals of Australia moved to the Australian subcontinent, cows were not domesticated across the human population, which means that their variation, they did not experience the same selection pressures, which means you will see most Aboriginal individuals won't be lactose tolerant because they don't have the gene for it, because they literally, until about 200 years ago, when colonizers came over to Australia, they've been geographically isolated. So they did not domesticate cows at all. But that's one example of it. Island and red hair, yeah. Or why blue eyes is more common in Swedish populations. I mean, again, it all depends on things that are basically humans have had this for a while. But again, those aren't exactly founder effect, because founder effect is not, the founder effect is there is isolation. The people who are isolated have taken a sample of alleles that is a sample of the alleles that were present at the time, and then they will go and develop their own thing. So actually, both of these examples are probably the founder effect with a little bit of natural selection brought into it because they didn't have the same selection pressures. So again, you can only see the founder effect instantaneously after. Actually, one example of the founder effect is probably if you took a sample of people at your school and you looked at their grades are going to be a representative sample of, they're going to be included as part of the sample of the larger population, but they might not be representative of the sample of the larger population. For example, if you maybe go to a grammar school, your grades might be higher, than just the grades of the general population, but you are a sample of that taken out and excluded into your school. So that probably would be a more real life example of it. So if you take, it's slightly different. Okay, give everyone a minute's comfort break, just because I need to get some water as well. And then we'll start classification. I don't know if we'll be able to get through biodiversity because I do want to stop at the end for questions again. But that's kind of natural selection and variation. We'll maybe do biodiversity in a later lesson. Again, if you have any questions, put them in the chat and I'll try and answer them before we start classification. Right. Right, slight comfort break over. Hope everyone has had some time to digest the content that we've covered. Classification. So it's kind of related. How can we classify organisms? I'm sure you guys already know this. Let's take a second to think about it. We can classify organisms. Well, we can classify organisms based on their similarities and differences. Now, remember, these don't have to only be observable similarities and differences now. We've got cool technology that helps us classify organisms based on their genetic and molecular similarities and differences. But back in the day, when they didn't have that, we can classify them based on their visual and literally from seeing them. When I look at an algae floating on water, I can immediately tell you that those two, me and the algae, are different, which means that me and the algae are probably not going to be classified in the same category. Organisms that are very similar are often classified into the same groups and subgroups then. For example, we all, I'm hoping, I can't be certain because, you know, everyone has their camera off and there'll be some people watching this recording on the YouTube, but hopefully everyone watching this video, we're all humans and we know that we're all humans. And that basically is a group that we've classified ourselves into. But we are also part of a larger subgroup with all the other mammals because we know that we didn't come from an egg. And so we can all classify ourselves as being outside of that subgroup. So we're all within different groups, within groups, within groups, basically is the idea of classification. And taxonomy is the study of classification, not to be confused with taxidermy, which is the process of stuffing dead animals with formalin and keeping them, kind of making them look as lifelike as possible. Lauren, yeah, we did actually all come from an egg. Yeah, good point. Yeah, I mean, I meant like a giant calcified egg, but yeah, we did actually all come from an egg as well. But what is the definition of a species? this should be something that you all know from your GCSE year. Examiners often report that people misunderstand the definition of species. A species is a group of individuals that have the most in common with each other. But what's more importantly is two individuals belong to the same species if they can successfully mate with each other. And actually, I believe that this definition is lacking something. If they can successfully mate with each other to produce fertile offspring. And then individuals in the same species tend to have similar appearance, anatomy, genetics, phylogeny, and physiology. But the most important thing is that they can successfully mate with each other to produce fertile offspring. And the reason is because horses and donkeys can successfully mate with each other to produce the mule, but that doesn't mean that they're in the same species. We've seen I'm sure people have seen images of the liger. It's a lion-tiger crossbreed. And again, that's something that can happen, but that doesn't mean that the liger the lions and tigers are the same species. Yeah, so as you can see, the offspring that produce of organisms within the same genus are often infertile. For example, a horse can be mated to a donkey to produce a mule, but mules are infertile. Now, interestingly enough, from the study of evolution, this is also becoming slightly contentious because there is a potential that a lot of us as humans, modern humans, have actually come from a hybrid of two different species that we thought couldn't have done this before. Homo sapiens and Homo neanderthal, there is scientific evidence to prove that they mated with each other at many different points 250,000 years ago, which means, and their offspring had to be fertile because we can detect those genes present in our population today. We can detect Neanderthal genes present within our population. So that implies that this definition might not be exactly the one that in 20 years' time your kids are being taught at school, because this definition is definitely going to be subject to change. Courtship behaviors. So again, you've probably seen this on like loads of David Attenborough documentaries, but courtship behaviors are ritualized actions to attract members of the opposite sex of the same species. Basically, they help create the bond that is needed to raise offspring together. And every species has a unique courtship ritual. There are also similarities in courtship behaviors within members of a genus. But what's really important is courtship behaviors is a form of reproductive isolation. So actually, two populations of the same species can actually have different courtship behaviors. And that's a form of reproductive isolation that can then lead to speciation kind of further down the line. So very important to note that. And again, I can't think of any real life examples of this, but having different courtship behaviors is not necessarily a marker of a species. Courtship behaviors promote prezygotic isolation. Basically, prezygotic isolation is breaking the series of barriers that prevent, prezygotic isolation is creating a barrier that prevents sperm and egg of one species to combine with the sperm and egg of a different species, because again, that would go way too far. And it is important as it ensures that when two individuals mate, the chances of a successful fertilization is increased. Binomial naming is really important. So binomial naming is genus and species. Genus is capitalized always, and then species is lowercase. It's usually written in italics, but the best way to, or in bold, and the best way to do it is just to put an underline underneath it. But yeah, so Canis familiaris is the modern domestic dog. Canis is the name of the genus, and familiaris is the name of the species. And again, it distinguishes the domestic dog from other members of the genus Canis. So Canis lupus or grey wolves, Canis aureus or golden jackals, and Canis latrans or coyotes. Examples of different Canis species organisms. Okay, phylogeny. Phylogeny is basically the study of genetics. It's pretty cool. It's the study of family trees, actually, specifically. So phylogeny is the study of evolutionary relationships between organisms, and this can be done through, basically, it's allowing for classification of organisms based on 8 taxonomical groups. This is the taxonomical hierarchy. So all of life is the main one, everything that's living. That's defined by Mrs. Gren. So I actually don't remember what Mrs. Gren stands for because I haven't been examined on it in years. But Mrs. Gren indicates that something is a living organism. Then you have domains, kingdoms, phyla, class, order, families, genuses, species. The best way to remember that is the acronym, did King Philip come over from Great Spain? Or did King Philip come over for Great Soup? Again, it's the best way to remember kind of just the order, the taxonomical order. There is no overlap between groups. You can only be in one group at each level. And higher groups have much more organisms. Lower groups have much less organisms, but the lesser number of organisms are much closer in relation to each other. And yeah, so organisms in the lower groups are more closely related to each other. And the classification system is continually evolving and changing as new types of analysis are discovered, like gene sequencing. So domain, we'll talk about this in more detail, the 3 domains. I'll quickly, just at this point, take a brief detour to talk about how we've discovered all of this, because it's very interesting. So if you start at the beginning, you look at organisms, how they were classed years ago, that was through visible characteristics. But we then discovered something called convergent evolution. Convergent evolution is where organisms that aren't very closely related to each other will develop similar characteristics because they have similar environmental selection pressures. So, for example, bats are, although animals, they're mammals, they're winged and have echolocation, which are different skills that have enabled them to survive and get food when they need to. But that doesn't mean that they're suddenly birds. Dolphins underwater, they've got flippers. They're mammals, however. So there are different studies that we've now run that basically help us classify organisms by their group. So we now run genome sequencing. The first one we did was the Human Genome Project, which was hugely successful. It mapped all of our genes. It's very cool. I don't know if you've seen the map, Human Genome Map. It basically, this is kind of every single one of your genes and every single one of your locations of every gene. And we now know that. We know what every gene does, sort of, and people do more research into that, but we kind of know what every gene is in the body, which is pretty cool. But yeah, genomics is one way we can do it. We can do it still through anatomical, visual, kind of, that gives us evidence. It's very useful still. We can use, specific protein sequencing. So one of the proteins that we use is hemoglobin, because hemoglobin is found in basically every animal organism. So within the animal class, it can help us split all of the animals down into what their kind of constituent genes and constituent proteins codes are. But again, it's less specific than gene sequencing. And then the last kind of big one that we can kind of do is we can do it through embryological sequencing, because embryology is very interesting, because as organisms develop, they kind of look like different things because you develop in a particularly predicted way based on gene planning and gene maps, which are through things like Hox genes and homeobox genes. Again, that'll be a concept I cover in you'll cover in A2, but I'll cover it in next semester's lessons for those that are now doing their A2. But you've got your Hox genes, you've got your homeobox genes. And again, these kind of tell your body where to put things in body planning. So we can look at that and we can compare body plans to determine kind of what organisms kind of are more closely related to each other. Okay, the last thing I'm going to talk about is the 3-domain system. 3 domains and 5 kingdoms. Oh, and then the last thing that this lesson would be talking about is phylogenetic and genome sequencing. I'll kind of skip over that then. So 3 domains: bacteria, archaea, and eukarya. So bacteria are all the bacteria, they're plasmids, 70S ribosomes, peptidoglycan cell walls. Archaea are unicellular prokaryotic organisms that are similar to bacteria, but also relate to like algae. And then eukarya is all of us, eukaryotic, so plants, animals, fungi, everything that is eukaryotic. 5 kingdoms. So the 5 kingdoms are prokaryotes, which are unicellular, protocysts, which are unicellular protists like amoeba and paramecia, fungi like mushrooms, plantae, which are all your plants, and animalia, which are all your animals, basically. So each kingdom can be broken down into different phyla. So animals have 11 different phyla, but again, you don't need to know that. And then a phyla is broken down into a class into order, order into family, family into genus, and genus into species. Kind of makes sense. Okay. So the study of taxonomy relies heavily on the field of phylogenetics. Phylogenetics are a series of techniques used to find similarities between organisms during their classifications. And you have different advances in immunology and genome sequencing. So antibodies are specialized proteins which recognize and bind to antigens. Again, I'll talk about this in more detail when I do the immunology lecture, but antigens can vary between species. But antigens with the same function can have different structures in different species, which means that antigens can then be used to compare species. Pretty cool. And again, hemoglobin antibodies are used when comparing humans to animals because almost all organisms have hemoglobin. If the antibody binds, the organisms may be related to humans due to the structural similarity of hemoglobin. And then your last bit is gene sequencing, which is determining the entire base sequence and compare to other species and then assess the similarity of that. Kind of all makes sense. I've been asked one question in the chat. Can I expand on what protozoa are? So protozoa are very similar to prokaryotic organisms. They are still a form of eukaryote, but they're like your amoeba, your unicellular, small, very small living organisms that can exist on their own. But I'll show you protozoa. So they're microscopic unicellular eukaryotes that have relatively complex internal structures and carry out complex metabolic activities. Basically, they're designed to live by themselves. I'll show you a bit of a picture of them, what their internal structure kind of looks like, but they are nucleated. So you can see that they've got very similar structures they've got oral grooves, which again, bacteria won't have, and they've got nuclei, but they're unicellular nucleated organisms. Can I link where the slides and recorded lecture will be? Yeah. So if you go to, I will just put the link to the Study Mind YouTube channel on here on the chat. And we'll kind of wrap up with that. Paramecium, yeah, that is paramecium. Paramecium is a type of eukaryote, is a type of protozoa. Give me a sec. Computer seems to be quite slow. Yeah, protozoa. So paramecium is a very specific type of protozoa. Protozoa are a general term. And it includes amoeba. Yeah. So amoeba, oh, I will just put that to everyone in the chat so everyone can get it. That's a YouTube link. If you go under the tab that says live, all of the previous recordings will also be there. Any other questions? I'm happy to wait if anyone has any questions. What about protists? So protists are like parasitic organisms, but yeah, they're also protozoa. Protist is actually the singular of protozoa. Protozoa is basically the pluralic. So plural of protist is protozoa. If there are no other questions, I'm happy to wrap up for the session then. Hope that's okay. I'll still wait around. Could it be linked to could it be linked endosymbiotic theory? Well, in the sense that only in the sense that endosymbiotic theory and evolution are both about mutual benefit of species and individuals. But again, endosymbiotic is not an evolutionary form. It didn't provide it provided multicellular organisms with benefits. It helped us become multicellular animals, but it didn't really. It's not exactly the same thing. No worries. Catch you guys later then.